Let's welcome our last speaker. Mr. Aylwin Lin is currently a third-year PhD candidate under TIGP Molecular Medicine program. He graduated from University College London with Master of Pharmacy and was a practicing pharmacist in Malaysia for more than 6 years before starting his PhD in Taiwan. His research interest is precision medicine in complex diseases under the supervision of Dr. Cathy Fann from the Institute of Biomedical Sciences, Academia Sinica.
Good afternoon. Thank you. Good afternoon. Today I'm going to share the story of how we use unsupervised learning in complex diseases for precision medicine. So first of all, what is complex diseases? So complex diseases are diseases that are influenced by both genetics and non-genetic factors. Another complex nature of complex diseases is that they are very highly heterogeneous. So what it means is if you have the same disease, you have a disease, and the person next to you that have the same disease, you usually wouldn't behave the same and respond to the medicine the same. So we already know this, but medicine is practiced by a one-size-fits-all approach, where everyone with the same disease will be treated with the same medicine, but we all respond differently. So essentially, precision medicine is about giving the right drug to the right subgroup of patients. So how can we make complex diseases less complex for precision medicine? I'll give you a 1-minute crash course on genome-wide association study. So we'll start usually by biobank, where data is collected. Individuals will usually be genotyped by array or whole exome sequencing or whole genome sequencing. Then they can be assigned ancestry by this genetic principal component. Imputation is a statistical way to essentially add in more genotype data. And then genome-wide association study, which is basically a multivariate logistic regression across around 10 million single nucleotide polymorphism across our DNA. And then subsequently, we usually will perform post-GWAS analysis as well. So for my study, we are looking at something called metabolic syndrome. So metabolic syndrome consists of diabetes, hypertension, obesity, dyslipidemia, and metabolic syndrome essentially any individuals that have 3 or more of the criteria will be classified as metabolic syndrome. So in our study, firstly, we extracted individuals from UK Biobank, which is around 334,000 Europeans. And then we applied unsupervised clustering, or K-mean clustering here, using the features of metabolic syndrome. Then we managed to identify endophenotypes here. And then we compared the phenotypic comparison across 87 quantitative traits. And we also look at 25 clinical outcomes, the association of 25 clinical outcomes. And subsequently, to identify the genotypic traits, we perform genome-wide association study of all the endophenotypes. And using the GWAS results, we then did genotypic comparison, and then precision drug repurposing, and also precision risk prediction. And Taiwan Biobank with 127,000 Han Chinese is used as a validation cohort. So we identify 5 metabolic syndrome endophenotypes. So the first one is a non-descriptive metabolic syndrome, which is non-descriptive across 87 quantitative traits, with traits more similar to pre-metabolic syndrome and healthy. Cluster 2 is defined by hypertension with the highest proportion of female. Cluster 3 being obese. Cluster 4, lipodystrophy-like, with highest triglyceride, LDL, low HDL, and yet relatively normal obesity trait, whereas cluster 5 is defined by hyperglycemic. We also noticed that the 5 different clusters have similar proportion across the 2 biobanks. So for any endophenotype to be clinically relevant, they should have differing risk. So we look at 25 clinical outcomes, which include cardiovascular diseases, dementia, chronic kidney diseases, and various types of cancer. Here I'm just showing one of it, which is composite cardiovascular outcome in UK Biobank, which is the combination of cardiovascular diseases, coronary revascularization and mortality due to cardiovascular diseases. So from here, we can kind of see that the different clusters do have differing risks. For example, cluster 5 had the highest risk before adjusting for type 2 diabetes status. Cluster 1 had the highest risk after adjusting for type 2 diabetes status, whereas cluster 2 had the lowest risk, while cluster 3 and 4 had similar risks. So then we try to look at a relatively similar trait in Taiwan Biobank, which is self-reported coronary artery disease. Again, we see the same thing. Cluster 5, highest risk before adjusting for type 2 diabetes, and then cluster 1 after adjusting for type 2 diabetes status. This slide was the one that I struggled the most with because I have to summarize 12 GWAS results with post-GWAS analysis, which consists of more than 40 figures and tables. So please read the preprint here. So I'll just highlight our key findings. So when we overlap the genes that were associated with each of the clusters, we've identified 3 overlapping genes, except for cluster 2, which is LPCAT2, NUTT21, and OGFOD1. And LPCAT2 is actually a novel discovery, which is highly expressed in immune cells, probably highlighting chronic inflammation as the shared pathophysiology of metabolic syndrome. Whereas cluster 1 is quite interesting because it is highly cardiovascular disease risk. So we identified the genes in cluster 1 that are uniquely expressed in cardiomyocytes and also skeletal muscles. Whereas for cluster 5, most of the genes identified are well-known type 2 diabetes genes. However, something to note is that the sample size of our cluster 5 is actually quite small, around 3,000, compared to GWAS of other type 2 diabetes. So when we do GWAS of case control, usually homogeneity of cases matter more than the size. And then we noticed something interesting as well. Overlap between cluster 1, 3, and 4 are genes that are expressed in the brain, especially hypothalamus and pituitary gland, indicating the role of neuroendocrine system in metabolic syndrome. We also identify some of the genes in Taiwan Biobank. So next, precision drug repurposing. So a coding gene can be translated to a protein. And if a protein has a drug target, then we can put this link. So I'm using this bioinformatic tool called GRAB, which is essentially using GWAS results or genes from GWAS result and then linking it to a drug target. So from here, we notice that the different clusters do point towards different drugs. So for cluster 1, it was drug targeting the cardiovascular system, especially antihypertensive drugs. Cluster 3 was anticoagulants and anti-obesity agents. Cluster 4 was lipid-modifying drugs, and cluster 5 was drugs targeting the GI tract and metabolism. And then the next part, in genetic epidemiology, we always like to look at polygenic risk score. So what is polygenic risk score? Polygenic risk score, according to my professor, is like looking into the crystal ball to predict the disease based on your genes. So here, but most of the time, when we predict disease based on polygenic risk score for complex diseases, they are usually not as successful because they are highly heterogeneous. So here, when we did endophenotypes, this one is using the original sample size, we can already see that cluster 4, despite having smaller sample size, has better accuracy here. Whereas, so mostly implied that cluster 4 is highly influenced by genetics. So to reduce the impact of sample size, we randomly sampled 3,800 cases from each of them, and 3 endophenotypes performed better than the metabolic syndrome here. Cluster 1 performed similarly, and cluster 2 performed less well. So here we managed to show that we can increase the polygenic risk score accuracy by decreasing the heterogeneity of metabolic syndrome through clustering. So in conclusion, we managed to apply novel methodology from machine learning, genetic epidemiology, and statistical genetics to identify clinically relevant endophenotypes. And yeah, that's all for my presentation today. And I would like to thank my professor and my lab members from my lab. And if you are interested, you can always scan this QR code for the preprint. Thank you.
Thank you, Aylwin.